All right, we should get started. Welcome, everyone. Welcome to the Society of Surgery of Alimentary Tract webinar. I'm Vikas Tudeja, HPB surgeon at University of Alabama at Birmingham. I'm co-moderating this session with Dr. Emily Winslow, professor of surgery at University of Wisconsin, and she's also a liver pancreas surgeon. Today's webinar is focused on team science. As we all know, the key to scientific success and high quality research today is collaborations and doing research as part of a team. In today's webinar, we will focus on how to create and nurture a high functioning team. We have an amazing lineup of speakers. There's a slight change. Dr. Saj Khan, who is from Yale, will go first. Dr. Saj Khan is a hepatopancreatobiliary surgeon and conducts high-level translation research. He will introduce us to the concept of team science and go over very important salient features. Saj.
Okay. Okay, thank you, Emily and Vikas, for moderating this session. It's actually an honor to be speaking with such great presenters here and some very big national figures in surgery. So over the next 15 minutes or so, I'll try to talk a little bit about team science in translational research, and I thank the SSAT for allowing me to speak here. So whenever we start with a research project, this is the way I think about it in the beginning, and research projects can often seem like a long, windy road with no end in sight. However, no one can walk that road alone anymore, and the NIH has realized that. Scientific problems have become more challenging given the vast amount of information and technology that exists across the globe. Because of that, we've seen over the years that there are more NIH-funded research projects with multi-author papers, and the greater number of authors are actually associated with a greater citation influence as well too. And this is just a study I found from Science not that long ago, bringing home that point. So before talking about team science, one needs to understand what is team science. So team science is a collaborative effort to address a scientific challenge by leveraging experts in the field. So one needs to have a compelling, clinically driven research question to start. So the team is comprised of various individuals. The team addresses a single research project, which is clinically focused. A team is comprised of members with training and expertise for multiple scientific disciplines. And the team works together to combine and integrate their knowledge, skills, and their perspective from different fields. And the reason that this approach is important is because it enables society to solve complex problems in biomedical sciences, and one can argue in surgery and various other disciplines in medicine as well. So, over the course of the remaining talk, I'm going to provide some suggestions of how to apply team science to translational scientific research, and give an example of a project that I was fortunate to be involved in with a very solid team. So the first thing that I would say is it's important to assemble a winning team. And one important thing before one assembles the team is to actually have a question that needs to be answered, because the research question drives who you need on your team. Quality research needs a quality team. No one can do it by themselves. And the team leader needs to identify the right people for their team. And this could include multiple people. This should include key collaborators. It should include mentors and advisors. And it can also include trainees and staff. Sometimes biostatisticians are very important for translational research, where large volume of data needs to be interpreted sometimes, and it's hard to do sometimes on your own. Obviously need some lab staff and some lab space sometimes to do this, or collaborators, lab space. And our trainees are critical. And not only do we train our students to become residents, or residents to become attending surgeons, and fellows to become junior faculty, we have students, pre-docs and postdocs, that often are critical for this winning team. After a team is created, it's important to create an environment for your team to succeed. And this environment should stimulate their interest and the interest of the entire team. And it's important for the PI of a project, for example, to provide guidance, develop and mature research questions, to continue to motivate that team, dedicate time to that team, and learn from your team. So a lot of it is investing in those around you, and in a certain way, I think that some of those concepts are servant leadership for some of our leaders in the audience. So the question that I thought, just to use for as an example, is colon cancer liver metastasis. Colon cancer liver metastasis is a clinical scientific challenge. And one particular area that my lab is interested in is studying the laterality of colon cancer liver metastasis based on the primary tumor. So to put some perspective on it, so different clinical behavior of right-sided versus left-sided derived colorectal cancer liver metastasis is something that we know about a lot from clinical studies. There's poorer survival from patients with the right-sided derived colon cancer versus left-sided derived colon cancer. And that's not just for patients with non-metastatic disease, it's also the case for metastasis that have developed distant metastasis as well too. And this is strongly suggested that there's a difference in biology when you control for the stage, et cetera, because we also know from literature, from studies in the last several years, that patients that with right-sided derived metastatic tumors tend to be less responsive to EGFR inhibitors. And most of the studies in recent years have focused on genomics and transcriptomics of the primary tumor. So what we were interested in understanding is, are there biological drivers in the metastatic tumors? And we wanted to look at extensive genomic and transcriptomic differences. So this is a complex problem that requires a lot of expertise in a team in order to help address the research question. So the question that we asked our team is, what are the metabolic and transcriptomic differences between right-sided and left-sided derived colon cancer liver metastasis? Our study conceptualization was focused on metabolomics of liver metastasis. So metabolomics is something that my lab has a special interest in, as does my team that I get to work with every day here in Connecticut. Metabolomics is untargeted quantification of small molecule metabolites. And what we were interested in doing is using metabolomics data and integrating it with transcriptomics data. And we were focused on liver metastasis to address our research question. And the reason for that is metabolomics, number one, is a bit understudied in actually liver metastasis, and there's some clinical relevance, as our background showed before. So in order to have the research project going, so to put some examples of how we address this, first step was to identify key collaborators and work with the research mentors. Because as the late Steve Jobs had once said, it doesn't make sense to collaborate with smart people and then tell them what to do. We collaborate with smart people so they can tell us what to do. So I was fortunate enough to have prior mentors and collaborators here at Yale that have helped so much in this project. So Pat Patey is one of the super smart surgeon scientists from Memorial Sloan Kettering. I've been fortunate to have him as a mentor from some of my prior training. Caroline Johnson is a metabolomic expert I get to work with every day at Yale, and June Liu is a RNA expert who I get to work with at Yale, and actually was my scientific mentor at one point, and now has evolved into a collaborator. So in order to succeed in this kind of a project, we engage with our trainees. And I've been fortunate enough to get to work with two of these individuals on this slide. One is Montana Morris, who's actually who was a medical student at the time of this project. He's a general surgery resident now at NYU, and I heard doing great things. And Abhishek Jain, who's one of our postdoctoral fellows here at Yale. So what we decided to look at, or Montana and Abhishek decided to look at, is surgical patients that have had surgery at Memorial Sloan Kettering, and 90 patients were examined. And we were fortunate enough to examine patients' liver metastasis and normal liver samples with the cohort listed below. And using these fresh tissue from tumors in normal liver, we perform mass spec experiments in the Johnson lab. So Dr. Caroline Johnson is one of my key collaborators, and we performed RNA sequencing experiments at our core facility here. And we were also able to use the GEO data set, which is a very rich way to examine data and get validation studies. For a lot of people are doing that across the globe. And so we took patients' normal liver and liver mets and ran mass spec experiments on them. We separated the group into left-sided versus right-sided derived liver mets. and we determine which metabolites are associated with the laterality preference. And this is just the cohort of the entire group, not just the patients that have had, for which we had tissue. And this just brings home the point that patients with the right-sided derived liver mets have a poorer outcome than left-sided derived liver mets. And this is where it starts to get a little bit more exciting. And again, this is all work done by members of the lab. It's just something that I helped guide, but I got to work with members in the lab and our team. But the first thing that we found is reactive oxidation species favor right-sided derived liver mets. And I'm not going to make this into too much of a scientific talk, but when one can see cysteine, ascorbic acid, and glycodeoxycholic acid, which are reactive oxidation species associated metabolites, are overexpressed in the right-sided derived liver mets, specifically the liver tumor compared to the left-sided. Interestingly, we found the same thing in regards to bile acids. And just keep in mind, these are not actually colon tumors, these are liver mets tumors. And we found that right-sided derived liver mets actually had an overexpression of bile acids in their liver mets compared to left-sided. And carnitines favor left-sided compared to right-sided. So using this metabolomics data, what we next did is RNA-seq experiments, and Vadim Kurbatoff is a PhD candidate here and doing great things as well. And what he did is he did RNA sequencing experiments, and what he found, which you can nicely see on this heat map, is that patients with right-sided derived liver mets have a more similar RNA makeup compared to those with left-sided derived liver mets. When we did a supervised gene expression analysis, we found actually this gene called SERCAM, which is important for metastasis, is overexpressed in right-sided derived liver mets compared to left-sided derived liver mets. And when we did an IPA analysis, this is basically a way to put all the data together and mix the metabolomics data and look at it in relation to the transcriptomic data. We found that a very important gene pathway, TGF-beta, was central to this entire network and potentially is driving this process. And again, at this point, that's what our metabolomics and transcriptomics data shows. And TGF-beta is important for multiple different reasons, but it also is important for the PI3 kinase pathway and MECR signaling. So that was pretty exciting in and of itself. And so what we did is we again used our team, and we actually submitted this to our team over here submitted this to a journal, and the journal actually said, hey, you have to show us a little bit more to make this more than an association. And what we did next is we did immunostaining, and Bashi's son here, who was a research scientist that spent time with us as well too, did a very nice, elegant experiment with immunostaining. And he specifically looked at some of the genes on the right-sided compared to the left-sided. And what he found is that TGF-beta is overexpressed in right-sided tumors compared to left-sided. And SERCAM is also overexpressed in right-sided compared to left-sided. So this is not a talk so much about the details of the science, but just to promote how team science can enable us to answer tough problems in science. So in summary, team science enables society to address complex scientific challenges, and I use colon cancer liver mets as an example. Team science can address clinically driven research questions, and our question was, what's the differences between right-sided versus left-sided derived liver metastasis? It's important to identify the right individuals for your team, and they can include mentors, collaborators, trainees, and multiple other people in your social circle. And finally, it's important once a team is assembled to enable your team to succeed. And I use this example of identifying key metabolic differences between right-sided versus left-sided derived liver mets to show that to be the case. So again, the work that was done here is the teamed approach, and there's multiple people from Yale and from Memorial Sloan Kettering, and obviously funding that enable this work to be done. So, and that's all I wanted to say. Emily Vikas, thanks again.
Great. Yeah, thank you so much for that really nice talk. And I think you can see always from the last slide, the more the merrier, right, in general. And that's what we're here to talk about today. So next, Dr. Heather Yo is going to talk about creating the research team, which you emphasized. And then we're going to go back to talking about team science in different other environments. So thank you for that nice introduction and first talk. So there you go. Great, Heather.
Perfect. Thank you so much. Thanks, Dr. Winslow. Thanks, Dr. Dudeja. And thanks to the SSAT for planning this webinar. Also, Dr. Khan, it was great to see your example and definitions of team science. I'm going to back things up a little bit and talk just kind of about how you even start building your research team. I can move my slides. Yeah, sorry. All right, so I'm going to talk about assessing your needs, how you interview and hire, how you think about and find effective team members, how you manage that work. And then I'm going to just give an example of our lab and show you what we've built in terms of a website and social media to help kind of move that forward. Sorry, I'm struggling with the pointer. All right, so what do you need? Each of these different projects are very different, right? Like I'm a kind of clinical outcomes researcher, there's translational research, but there are common members of surgical teams that are really important to help build them. Research coordinators are important. Having someone to help you with regulatory support is important. Statistician, methodologist, or whoever's going to help you with your data analytics. Having a research nurse if you're going to be doing some translational research that involves patients, as well as already mentioned here, research fellows, students, PhDs, and kind of the people that are going to do a lot of the bulk work of your team. Why can I not get pulled? But it's a wrong arrow. I'm sorry about that. So what do you need? It's really important to kind of get a sense of what you need for your team and how much space you have. I live in New York City, where space is a kind of prime thing that we fight over. So you have to understand how big your team is and whether or not you're going to need desks. If you're doing patient-oriented research, you're going to need locked cabinets. You need to find conference rooms so that you can have meetings. It's gotten a little bit different now that we do a lot of these meetings via Zoom. So you can actually use a little less space, but space still matters. Equipment, it's important for any kind of studies or research that you're doing that you have computers and software that you can use to kind of move your data forward. And that may be different if you're doing translational work. Well, you'll need a lab and a lot of lab resources, but understanding kind of what equipment or resources you need to get your labs started are important. You have to think about how you build support. So you need protective funds, protected time, startup funds, access to data, and or lab resources. It really depends kind of what the lab is that you're trying to put together. I want to give some really practical tips. So when I started doing research, I was really excited about anyone that seemed interested in my work and wanted to start joining my lab. And I found that I was intermittently having people who were more or less motivated. And so I think it's really important that as you start to build your team, you think about actually how you're going to interview and hire. And so part of that is understanding who you are as a researcher and who will complement you. We actually created an interview guide to try and make sure that all our candidates are examined by the same measures. So we basically, we just hired our most recent research assistant. She is a rock star. And we actually got, when we posted online on the internet and locally, and we got over 100 applications. So we started by screening with phone interviews to just get a sense of the candidate and see who fit with the kind of overall interest in the research that we were doing. And then my research team actually had them, we assigned them an active task. So we had them choose an article, any article that our lab had worked on, and make a presentation to see if they actually could understand the research and understand what we'd done. And also, we had them perform a mock patient consent. So imagine they were enrolling a patient for one of our trials. The other thing to keep in mind is that, so we did hire one person out of the 90-some resumes that we got, but it's important to kind of keep those people on file, because as you look through those CVs and resumes, these people are also potential collaborators and people that you might reach out to in the future if you need additional help. So how do you actually find those effective team members? For us, I do a lot of team research, and a lot of the research I do is not with surgeons. I do research with geriatricians, I do research with pediatricians, gastroenterologists. So I think it's really important that you attend seminars and lectures at talks that are outside of your direct profession, but of interest to you. So we go, you know, I'm a joint appointment in our population health department. And what that means is I go to most of their grand rounds and conferences, and people that are doing research that I think parallels mine are people that I set up meetings with and try and spend time with. I think that also a lot of hospitals and universities have additional statistical support, and you can learn some of the new statistical techniques by attending different biostatistical lectures. So we do that as a team. I also think it's important to use your network. As we all go to academic meetings, touch base with people in the clinical trials office to see what people at your institution or local institutions are doing work similar to yours. Go to academic meetings, SSAT, for example, SSO, go to these different meetings and attend people's lectures who are doing research similar to yours and who may be future collaborators for you. Talk to your med school classmates. I actually have someone who was not a med school classmate, but someone that I did a master's with some years ago who's doing some qualitative research with me. And then look on social media. Social media is an excellent way to see people doing research in your field and to build potential collaborators through that mechanism. It's always important to figure out someone's interest and ability to collaborate. So I work a ton with medical students, and I'll show you a link to our research website, and you'll see that I have probably on average about 4 medical students at all times working in our lab. So I think it's important to understand their capacity, because a M1 who's just in the books trying to learn anatomy probably doesn't have as much time as a med student who's on a summer break or even taking a year off for research. I think that it's important that early in your data, you talk to statisticians and or methodologists to make sure that you're doing your research right from the very start. And then once you do start figuring out your mentorship and trainees, I think it's important that you actually set time for them, dedicated time into your schedule, and get goal setting conversations very early in the parts of them joining your team, so that you can make sure that they're getting what they need out of the relationship and you are getting what you need as well. Once it comes time to managing the work, and this has changed a little bit with the since the pandemic, I think it's important to figure out ways to set your research goals. So a lot of people that work with me will tell you that I'm an idealist and a person that has a lot of ideas, almost flight of ideas. When we talk about one topic, I'm like, oh, that's a great idea. We should also study this. So it's important that you pair yourself with people that can match with you. And so I do really well with people who are super detailed and like putting lists together, because they can keep me focused and on track. So my lab meeting always has an agenda. There is time for kind of free flow, but we try and get our agenda done and then have time where we kind of brainstorm and free flow towards the end. I think it's important to block time for research for you during your clinical practice. We all are getting busy in our clinical practices, and it's important to actually find time to focus on that. Keep a running list of your projects. I actually have a spreadsheet that we look at when we have our meetings, where we talk about what the roadblock is, what the status is, where we are aiming to publish, what our funding target is, and who the point person is for moving the project forward. I think, as I mentioned, things have changed specifically. So we do actually a lot of remote teams. A lot of newer, younger people that are joining teams do like the flexibility of sometimes working home. So we, and in New York City, transportation and travel can be very difficult at the times of day that a research team might meet, super early in the morning or the evening. So I'm trying to be really mindful of where my team is. I also, as those who know me know, is I'm a little bit of a talker. So I really try to find time where I ask a question and listen. It's really important in team science that you leave room for questions with your trainees. And as mentioned, some things have to be done in person, for example, patient enrollment, but a lot of stuff can be done virtually. So I'm just going to end with this. I think it's important to try and engage your trainees, think proactively about your team building, make sure that you provide contact info for collaborators. So virtual calling cards or QR codes with your team's information are great to have when you go to academic meetings. We just put a lab website together. If you want to check it out, it's called yoresearchlab.org. And it really just has information on how to collaborate with us, what work we've been doing, what our trainees have been doing. And I really enjoy working in a big group of people. So team science is great. And thanks for having me.
Thank you, Heather. Next, we have Dr. Joe Hines, who is the professor and chair of Department of Surgery at the David Geffen School of Medicine at UCLA and the William P. Longmire Jr. Chair in Surgery. He's going to talk about fostering an environment conducive to interdisciplinary collaborative research. I do want to mention to all the attendees that if you have questions, please type them in the question and answer box that is available to you. We will address all the questions from the panel in the end, so please keep writing questions. Thank you. Dr. Hines.
Thank you. Thanks for the invitation. It was good to hear our first two speakers, and I'm going to talk about how to create team science, kind of a departmental level, some of my experience with that, and start off by giving you some statistics as to why I think this is really a great way to go as an academic surgeon. So, we are all faced with all the missions that we have to accomplish every day, both individually and collectively in programs and departments. And research is one part of those. And we are all busy surgeons trying to complete our clinical work every day, and obviously have missions of education, community outreach with some of the work we do through the month. And so because we have such time commitment to so many different areas, and our services generally run that we're sort of always on. We don't have large swaths of time for months, as some of our colleagues say in the Department of Medicine have to devote to research. It's really important to maximize the possibilities you have. And I think by being part of the team, being part of a team project, you can really do that. So I'm going to give you some statistics just on funding, what current status is. These are some institutions that I polled as far as average. This is average NIH funding per PI at each institution. And the point of this is that many institutions, there seems to be, this is not just that the amount of funding is increasing. This is more around how funding is starting to be concentrated in individuals rather than more broadly across faculty. So for those who don't have funding, it becomes more of a challenge as you compete for those dollars. And this is a statistics about each institute and what their most commonly funded mechanism is and the success rate. So you can see those listed here. Gosh, if you want to apply to the NIA, I think that's looking pretty good. But generally speaking, funding rates are 20% or less for common R mechanisms. Yeah, but you can see there the list, and it's an interesting table depending on the area of research that you're doing in the institute that you target. This is funding success rate by U54. U54 are specialized center grants, which require team science collaboration, a lot of resources and cores that come together to make a U54. But my point of showing this is that when you work together as a team and you submit these team grants, the funding rates are generally higher across the mechanisms. And in fact, gosh, some of them very high. They're not funding as many of these projects because they're larger amounts of money. But there's a lot of work that comes together to put them together. And when it's done successfully, you have a pretty good chance of getting funded. And this last statistics slide is really looking at all sort of team science or program project grants or P50s. If you just pay attention to the green line, that's all of them together as a percentage of NIH funding by institution. And a fair amount of funding now at each institution comes from these sort of larger team grants, upwards of 50%, 15% at some of the places. So definitely think there's momentum just based on those statistics to try to work together and create teams and ultimately probably have better science and outcomes with the projects that you do. And then this is statistics on PO1 success rates, program project grants. These grants have cores and usually 3 or 4 sort of RO1s and one grant, and pretty good success rates. Maybe not on the first submission, but on the second submission, likely pretty much higher likelihood of getting funded. So I'm all about the team, and it takes a while to create the team. It's not as if you can, from day one, put a team together and say you're going to apply. There needs to be a kind of a coalescence and a thematic area of research to be able to put the expertise together and the track record to be able to put together a team project grant. And sometimes it works. We have several PO1s in our department. This is an award that was actually a combo award between the Department of Defense and the NIH on solid organ transplantation. And if you are at a university, which I think a lot of us are, there's a lot of advantages by being part of a university and a university health system that are listed here. Big clinical volume for translational research. If you're associated with a university campus, there's lots of people that you can interact with and create teams with that are listed there. And of course, the infrastructure for clinical trials and expertise at a university are really outstanding. And so it's worth taking a look across your university to see who you might be able to collaborate. Oftentimes, non-clinicians are really excited about creating projects because they have ideas, but they need a sounding board. They need your expertise. And it's important to create those so that the work that ultimately comes out of it is the most meaningful. So back to this grid, here we are, you're the individual. This is what you're trying to accomplish every day. How do you get it done? And it kind of looks like this when you're getting started. In fact, this is a 2D image. I think it's maybe for like 4D, this confusing as to what to do and where to start. But I think, as the point was made earlier by Dr. Yao, that it's really, really important to get organized. For me, this was the most important thing in any success I had to do in research, and that was to book in every week time to devote to research. In fact, our lab meeting, which we still have every Tuesday at 9:30 in the morning, has been going on for 25 years now, and we all join virtually. We're not together together all the time, but having a regular cadence where you're getting together with your team, with your lab, talking about results, talking about plans, talking about potential publications and grant applications like was described, I think is really, really important. If you don't do that, then what happens is the week goes by and then it's the next week and then it's 6 weeks later and you haven't done it. So I think this is a skill. Getting organized and managing your calendar is really, really important. And as was said earlier, it's really important to have a very strong research question. So we've been successful with a couple. We've now in our second PO1. This is one of the grants that the person who leads our lab, Dr. Guido Eibel, runs in collaboration with some of the other scientists on campus, looking at the role of high-fat, high-calorie diet and pancreatic carcinogenesis using a KRAS transgenic mouse that spontaneously develops pancreatic cancer. When you feed mice high calorie, high fat diet, they get big, large, a lot of intra-abdominal adiposity. They get earlier and more precursor lesions called pan lesions, and more of them develop cancer in a shorter period of time. So we're studying the mechanism behind this. Our lab has been very mechanistic, not very translational, really around the biology of pancreatic carcinogenesis. And we've been able to turn this around for a second PO1 too. So our lab's been funded for almost 8 years now through a PO1 mechanism. And it requires, each of these projects are in essence an R01 that's in there embedded. And then there are cores associated with it. There's a statistical core and an animal core. And so that really requires you to bring together that group. And you can use that structure that you've created where you're having a lab meeting every week to begin to invite others to come to the lab meeting, share results, and create that team and that goodwill between other investigators. So you start to share data, and over time, you will come to a place where we'll be able to put projects together. A nice thing about doing a program, a PO1 application is oftentimes people chunk that out and also submit R01s following that in case the PO1 doesn't get funded. So there's lots of ways to manage this and for the teams to continue to work together. Another project that I was involved with was the submission of a spore. We submitted and got close to funding, resubmitted, was not funded, but the process putting together this application was even maybe more important than the funding. Of course, we love the funding, but it got us organized. And you can see the list of people that are involved with this. People that are not only on campus, but outside of campus, that were, advisors on our internal and external boards. And the process of putting it together made the science better in the end, even though we did not ultimately land this spore application. It was a great process to do. I was in the nitty gritty with putting all the details together and putting, this was when, we had computers, right? We did have computers when we put this together, but I was very excited when it came together as a final document, and I was so excited about it. I had it bound, and so it sits on my desk, even though it wasn't funded. It's over 500 pages long, and I think this type of thing is the ultimate team science. So how do you create that culture among your team? You got to be sure that you're transparent with results. Try not to one-up each other. Try to really be focused on what's the scientific question. It's really not about promoting people's careers through this work. People's careers get promoted through the work. It's really about asking important scientific questions, understanding the goals of everyone. When you're going to do a group publication together early on, talk about authorship. Talk about how that's going to look so that people aren't walking on eggshells when you get close to submission and people feel uncomfortable with it. And of course, when people have a publication or they get a small grant, really celebrate that. Get together, have a beer if you like a beer or a cup of coffee, and try to announce those things across the department or the program that you're working in so that everybody understands the progress that's being made. And of course, what's the currency? It's numbers of publications, it's the impact of the publications, it's the impact of the science, and we all ultimately want that to translate into patient care. So to the extent that we all move in that direction, it's really important. I found when you work with teams, I like this term called kind persistence. So I think that to get people to coalesce around a concept or a project or a direction, it does take a kind of circling back and keeping things on an agenda, but all along the way being kind to everybody and understanding everybody's input. And for it, truthfully, that outcome will be better as long as when everybody's ideas are incorporated into what you have in your final product as an application for a team grant. Some of the things we're doing in our department right now just to make it easier for people to do research. We've got surgeons, they're so smart, they got great ideas, but gosh, they're so busy. So how do we make things efficient for them? We just finished a strategic plan for the department, and a lot of the initiatives are around research. A recent thing that we started was to have a voucher program for people who want to access core facilities or create preliminary data that ultimately going to use for grant submission or publication. We'll give them $10,000 to get that done if they don't have access to the money. We have begun to embrace the concept of using scientific writers or grant writers. I think that there are departments, certainly big departments in the country that you're competing with who have those people embedded. It does make a difference if your grant looks pretty, it's well organized. The person who's reviewing it thinks that you're probably going to be able to execute better than if your grant doesn't look organized. So I think there's an advantage now to using scientific writing. We have someone in-house, but we also have a voucher program for that. So we will allow people to go outside of the department. It's much less expensive rather than hiring somebody in-house, and oftentimes they're even more experienced. We've created a clinical trials group, and then really working hard on our contracts and grants office to make things as easy as possible so that when you want to create that team, that you're not the person that's going to have to be worrying about putting together the bio sketches or getting the letters of support, all the details that have to go into a big grant. So with that, I think it's always great to have a goal. Right now, we're into our Blue Ridge ranking a little bit. We're creeping into it. We have a goal to be in the top 10. The ultimate goal is not to be in the top 10, but it does give you it does give you direction and it makes you think about what it is you need to put together to create an environment for people to be successful with their research with whatever topic it is. It's broad ranging, like Dr. Yao relayed, but to make it so that people feel that they're supported, their time is most efficiently utilized, and ultimately you want to have the best science possible. One metric is the ranking. It helps everyone to orient in one direction. So I appreciate the chance to talk and just give a few reflections. I'm always available. This is my cell phone if everyone ever wants to call me. I gave my cell phone to the candidates for residency this year, and they started texting me right away. I was pretty surprised. But anyway, I'm available if anybody would like to talk.
Great, thank you so much, Dr. Hines. I really appreciated that, as well as I learned a new term that I think I'll steal, of kind persistence. So maybe that's what the candidates were doing as well when they were texting.
Right.
So we'll see. All right, next, we're really lucky also to have Dr. Susan Tsai with us from the Ohio State University to talk about team science as it pertains to clinical trials. So a different form of a team, but same manifestation. Take it away, Susan.
Well, thanks again for the invitation to talk. And again, I'm coming at this from a slightly different viewpoint, but leveraging off of what Dr. Hines was saying about all the different stresses that we have as a surgeon, which are very unique. What I'm going to focus on is how we can perhaps utilize the clinical enterprise in a way that can accelerate team science and actually building your team beyond the people who are actually on your personnel list or included on your grants or your publications. So actually Cliff Coe is here, I think last week, and gave a really amazing talk about quality. And part of that talk, as I was sitting through it, he talked about the Donabedian model of like the framework for evaluating quality and how structures then provide processes, and then that leads to outcomes. And I think some variation of this applies to research as well. Like, I think certainly we have to have significant infrastructure for research. Then that allows us to do different processes. And so for me, what I'm going to talk about is creating kind of the clinical framework or the structure, and then how we built kind of the team. And this is mostly at MCW. I'm at Ohio State now, but this is really going to be work that was done at MCW. And then the processes we developed around that, biorepositories, clinical databases, and then how that then led to research outcomes. So for this, again, just maybe starting at looking at the clinical framework and team building, we really thought that with, again, pancreatic cancer, kind of, again, pivoting off of what Dr. Hines was talking about, there's very kind of few cases. We really do have to learn from every patient that walks through the door. And the value of this is not only to the patient themselves, the research program, but also the clinical enterprise, if you develop a very concise and programmatic approach to delivery of care. And so to do this, I think the most important things were things that were somewhat not very interesting. Like, we had to come to consensus about definitions of stage of disease, and then treatment pathways, and then really what we thought were endpoints of interest. And I think sometimes we start at the end of that and then we work backwards, but actually having this framework was really helpful in providing a lot of structure for any future research that was being done. And that was implemented at a clinical level. And then it really actually in the end facilitated research collaborations. So when we talk about teams, I know we talked about specialty teams. For us, on the preclinical or like pre-laboratory side of things, maybe the thought is to start with patients and clinicians. We always think about that dynamic, but then engaging, as everyone has talked about, basic science researchers, translational scientists. And then in the end, for us, this is translating again back into the clinical setting with developing a very strong clinical trials team as well. So I know I'll get in trouble a little bit for this, but growing up in Ann Arbor, we always would talk about the team, the team, the team. This is something that is actually, I think, when you walk out of the onto the field at the University of Michigan, you see that at the very top. It's the last thing you see as you walk out onto the field for the football players. I have to say that the correlate of that is probably this quote from Woody Hayes, which is essentially that you win with people. So as we think about that, was the team at MCW that was kind of built around pancreatic cancer. You can see it's an exhaustive amount of people, and luckily they were not all on our personnel, because I'm not sure what kind of funding we'd need to support this many people. But we were really aligned around this goal, which is that we really wanted to learn from every patient. So once we had the clinical infrastructure down, what we started to think of was how can we develop processes to learn from every patient? For us, this meant developing a detailed database so that we actually knew what happened to every patient. We strategically aligned a biorepository with all that data. We developed derivatives from that. We shared those freely with our investigators. And then from all that analysis, we started to come back to from kind of the bedside to bench, and then back to the bedside again with clinical trials. So we developed this integrated biorepository. Oops, sorry about that. Mohammed Aldakek I'm sorry, this is advancing, but Mohammed Aldakek is a Syrian trained physician who was not practicing in the US, but very high level, and was able to kind of maintain our database, which was highly annotated. I'm going to try and go back again, but really, again, he helped to, okay, I'm not going to go back to that slide. I'm sorry. He helped to kind of identify and then track all the patients that we kept in our database. And then from the biorepository, we had a very lean group, but just basically one technician that went and collected all the blood samples, all the tumor samples. I participated in kind of the xenograft and organoid creations. We had a high-risk screening clinic and then a rapid autopsy program. So kind of soup to nuts, I guess is the saying, or nuts to bolts, or from beginning to end of the disease spectrum, we tried to kind of capture every element of that. And what we started to do, again, from building the clinical enterprise to having kind of a hive mentality about how we treated patients, developing the processes to develop resources for infrastructure, then we started to be able to ask kind of translational questions. And so we probably have all seen this in some patients that we've taken care of, that you can start with the chemotherapy. And what I'm showing you here is time on the x-axis, and on the y-axis, CA19-9 levels. So a patient who was started on a chemotherapy, obviously not having a good biomarker response, switched to another therapy and had a dramatic response. And then kind of how do we first predict an immediate response rather than having this 2-month period of ineffective therapy? And then also, what can we do to maybe understand treatment duration, or sufficient or insufficient therapy? So just kind of taking that first question, as you all do, I'm sure you read the literature, you're aware of kind of where the field is going, and in pancreatic cancer, there's been a push towards molecular subtyping, whether it's classical versus basal-like. A lot of very interesting work coming out of Toronto, as well as the University of North Carolina, looking at a correlation between treatment response and subtyping. So with that in the background, I happened to review a paper by Jen Janier, where they had basically simplified the RNA sequencing down to a very rapid classifier that looked at 8 paired genes, and it could be turned around in approximately 8 days using RNAscope. And that to me was very clinically relevant. And so after the paper was published, I reached out to her and I said, you know, we have some funding, philanthropic funding for a clinical trial. I think partnering with you, this might be an interesting trial. So we developed this clinical trial called the pancreas trial. I know that's not terribly interesting, but it's called the pancreas trial. And basically patients are at diagnosis biopsied. They undergo the purist profiling, which is this RNAscope profiling of these 8 genes. There's a very rapid turnaround with actually 5 business days. And then based on their profiling, they're assigned a therapy. And then they have repeat biopsy at 2 months to have pre and post treatment, paired pre and post treatment biopsies. And then we kind of assess their response. So the value of clinical trials, obviously there's some value to the patient. Certainly there's value to the clinical practice and enterprise. Many patients will travel for clinical trials, but certainly we learn a tremendous amount from clinical trials, especially in collaboration with scientific teams that we can ask multiple different questions. And so again, I think that has been the emphasis at the program at MCW, certainly developing not only one, but actually a myriad of clinical trials to fill a complete clinical trial portfolio so that patients that walk through the door, regardless of stage of disease, will have many different options for them. So in a summary, what's worked for me is really kind of developing this team or clinical laboratory outside the lab and built on kind of standardized clinical pathways to harmonize things so that although not every patient is on a clinical trial, you're getting as close as possible to harmonizing the care pathways, building that clinical team so that everyone is actually engaged and excited, that kind of hive mentality where everybody is pushing in the same direction, and then developing processes to really capitalize on the unique patients and the specimens that are coming through, and then circling back then to redevelop options for clinical trials. So this is the incredible team of people at MCW, a fantastic pancreatic cancer program there. And then I'm very lucky to work with a new group of people, a new team, so to speak. So happy to take any questions.
Great. Thank you so much, Dr. Steig, for that talk and for helping us kind of wind it back to the patient, which I think is the most important. So we just have a couple minutes for questions. And I think I'll start out maybe with a provocative question. And maybe Dr. Hines, if you can start, as this is kind of on your topic, but when we recognize the importance of team science and multi-author kind of, or at least multiple PIs contributing to a single work, how do we deal with the fact that the currency is publications and middle authorship is, discounted in large part? How do we deal with that on a systems level? How do we deal with that to motivate people to participate jointly and equally in an important project?
Oftentimes, thanks for the question. I think often when there's a team that's going to be successful in a team submission, the productivity out of each of the individual teams that make up the bigger team is usually pretty substantial. I think as far as output, as far as numbers of publications, I don't see that as a challenge. Maybe it's around the most high impact publication, the big message that's going to come out of the project. And I think that again, having that discussion early on, you could have co-first authors if and that can be noted. But I think that a lot of these team science grants, they have individual projects that wouldn't necessarily cross over. Like in that spore one I showed, there's a salivary a project, and then there's a sort of a radiology project. So they don't necessarily cross. But I just think when you have a team that's doing well, the productivity is sufficient to be able to support everybody that's involved. Yeah.
Thank you.
If I actually may follow on that, Dr. Hines. So the other issue is, and I'm asking you because, being the chair, you go through the promotion and tenure process for your faculty. Many times the question is how many senior author paper you have. So if you're doing a lot of team science and you may not be on, so how does that, how do you pass through that, and any recommendations with respect to that?
Well, so what does a university want for your promotion? They want their faculty to be a professor, ultimately, that has an area of expertise, a defined area of expertise. And when a new faculty, when you start off new with your first position, oftentimes you have this kind of basket of projects that you're working on, from fairly clinical project, maybe you are doing basic science research. But I do think over time that begins to narrow. Probably if you're successful in the lab with your basic science research, or you're doing health services research, that ultimately you have a very robust databases that you're accessing, a statistical core that you're using, you're having high quality publications come out, that you begin to kind of coalesce your project begins, your focus kind of coalesce about the most high impact work that you do. And so I don't think there's magic numbers to the number of publications you need. It's so variable too from one university to the next. But I think in the beginning, you're in your first couple of years, and maybe you're working with a more senior PI. You want to be the first author, okay? And then as projects become before, then you're the last author. And sometimes there's some uncomfortableness in that transition with if you're working with a senior author, but it's okay. In the end, I think the senior author, if they're a reasonable human, have to recognize that that has to happen.
Working with reasonable humans is a good goal.
Okay, yes,
Yeah.
yes. In a kind,
Great.
persistent way. Yeah.
Yes, better. Dr. Yo, there was a question to you from the audience about, I think this question is twofold. How do you assess motivation? But more importantly, how does the student trying to join your lab demonstrate motivation as part of their interview process?
Yeah, that's one of the reasons that we actually have them complete a task. Is that we want to actually do a little bit of screening by having them do a task. So that's why we say, go pick one of the articles that came out of this lab, describe to us what you understood from that. And you can tell a lot from their motivation based on how much effort they put into it and how much they understand the science. We had one candidate who actually went and found like a presentation that one of the lab members had presented and cited that. You can also actually, I think the other thing that we've noticed is not everyone has the same resources or the same background or training. So it's important as you're interviewing them to make sure they know we've assigned you this task. If you have questions about that, please feel free to ask us. We're happy to explain to you, give you ideas, motivation. You may not have, you may have never given a presentation before. We're happy to share samples. We just want to see that they're motivated. And I'm sure that everyone here has different ways that they assess motivation. I often will come right out and say, what are you interested in getting out of working with us? How can we work together? I tell every medical student that works with me that I've done projects before where I was personally interested and projects before where I was doing it trying to get a publication. And there's a very different way that you motivate for the work as whether or not you care about the actual scientific question. And so I tell them I will not take them on in my lab unless they think of a scientific question that matters to them. And we integrate that to the current projects we're working on. So I think they have to, one, care about the science, and two, show their motivation through as we said, the project or expressing motivation. But I'd be interested in how other people here assess motivation too.
Saj, you got, any ideas?
Sure. Yeah, I think I'd say I have some similarities to how Dr. Yo approaches it. One of the things that I had mentioned is spending time and meeting with your team. So if you meet with your team regularly, that can get you a sense of if they're interested in the project or not, and particularly early on in a project as well. And if I get a sense, if they're not putting data together or they're not asking in provocative questions that might take the project to the next level, even if they don't have the data, maybe it's not the right project for them and it's time to pivot to a different project. So that's something I think meeting regularly helps quite a bit. I also, before I start a project with anyone, I actually ask them to come up with 10 questions that they want to have answered. And then I help fine tune their question before they actually get started on the project, because that way they're interested in what they want to do. They're more likely to be motivated to what they do instead of me telling them what to do. So.
I have Susan, go ahead.
Yeah, I have a question for the panel and maybe specifically for Dr. Hines. I think we talk about these like large team science grants, and it seems like it would be lovely if it kind of organically kind of comes together. But sometimes it feels a little bit forced. Like you have investigators who they're interested, for example, in pancreatic cancer, but they don't quite like really intersect, and you're trying to put together a team grant, and you're trying to figure out how you can like force a square peg into a round hole. So just wanted to kind of get everyone's thoughts about how to navigate that.
I think that challenge was why we ultimately weren't funded with the SPORE. The terrific projects individually, but how did that really weave together? It wasn't quite clear. I think an institution or a department can help foster creating that direction between the teams by putting forth some support. Okay, you all want to work on this over the next 18 to 24 months. We'll give you some administrative support and we'll give you some dollars. Let's give you some dollars to try to, there may be some, preliminary data that needs to be generated between groups and between projects. But even though we ultimately were funded, and those projects individually were strong, I want to emphasize that it took many years before that for those projects to even come to a place where we thought that could be weaved together into an application. So the earlier you start, where people will actually begin to kind of go similarly, so that there is that rationale, that theme that's created, and maybe asking your department or the cancer center or whatever group that you're working with, if there's some support early on to make that happen.
I have a question for Susan. Susan, and anybody else, please chime in. But, like when you are having clinical research between multiple institutes, and then there's, of course, confidential information, specimen processing done at different places. How have you managed that, especially in light of the clinical trial that you are running?
Yeah, so I mean, I think most multi-site trials have a multi-site team. And so there are actually, again, processes around this to kind of safeguard patient confidentiality. For the research side of things, we're batching everything. So we just collect all the specimens and hold them at the single institution until we're ready. And then we'll ship everything all at once, because we're not actually doing that in real time. But for the clinical specimens that are being handled in real time, obviously that they have to be, transported per protocol. Is that what you're asking? But I think, again, the clinical trials office is the essential part to this, because obviously there's a lot of regulatory components to this. And you want to make sure that, everything is handled in a way that actually protects the patients, ensures the integrity of the research. And then also, there's, as you may know, there's a significant amount of billing that happens for multi-site trials. So all of that has to be kind of collated and taken care of.
I'll just add to that from a clinical research / I do a lot of work with large organizations like the AAMC and the ACGME. And so you do often need to coordinate with the CTA, the clinical trials offices, and or the legal office. We have DUAs between, I actually just had a meeting today between, I need to get some data merged from the AAMC and the ACGME and then sent back to me de-identified. So you have to I had a meeting with like all these different organizations to try and figure out the logistical components of having those conversations. Would it be easier for them to just talk to each other and then give me the data? That it may well be, but it's important, I think, as a team coordinator, that everybody who's vested is involved in those conversations to kind of make sure they flow and people understand the importance of the project or the importance of the work.
I have one question, and then maybe we can end the conversation. But Saj, I mean, I saw amazing metabolomics data, but many times you have a question where these kind of things are important, but you don't understand the technique or the data which is put out. So how do you, is any thoughts from anybody regarding how you translate that information in team science? Because it's not your area of expertise. How do you translate that information? And second thing is, how do you, and a very important one, how do you ensure data integrity, right? I mean, that what is being translated in the paper to the community, to the grants, is actually, like, what's the integrity of the data? Any thoughts on that? Anybody?
Yeah, I mean, I can start. So Yeah, so that's a great point. So, and I think that's where once you ask the question, you have to, I think it's good to have the right team of people that can help answer that question with the experiments that you want. So, for example, for metabolomics data, I decided to team up with the metabolomics expert and someone that really understands mass spec, and I understand it, but not to the level of a PhD metabolomics expert. So I think that ends up being critical in order to mine the data with both of our labs. But that goes twofold. So I think that I would say that some of the basic scientists that we work with also are looking for clinicians to collaborate with to make their research clinically relevant and clinically applicable. So it's not just some sort of esoteric kind of a way to ask a scientific question. So I think we have a lot of opportunity as surgeons to provide some clinical context to it that I think the basic scientists value as well too. And then, as far as the integrity goes, I think that's a huge point, right? Some of us probably saw recently there was a New York Times article about some of these challenges, and I think that's where presenting data in lab meetings or in university settings to others, allowing others to criticize your work. I think no one likes to be criticized, but I think that's where like NIH style, you get criticized and you learn how to have cleaner data, or you want pure data. You don't want to have anything by mistake or at all. So those are just a few thoughts.
I would just, oh, sorry, I was just gonna agree. Like, I actually had we took my last lab meeting, and we took about 20 minutes in my last lab meeting to read through that New York Times article and to talk about with down to the med student how important it is if you feel uncomfortable or concerned about data that you can ask the question and double check the data. I think that as we are advancing further and further in science, being able to show how you got your final conclusions is more and more important. So important to keep good documentation of the research and work that you've done and the data sets that you've used, and or the genomic studies you've looked at. I think that making sure your lab or anyone else who collaborates with your lab feels comfortable speaking up is super important.
Great. Thank you so much, everyone. We really enjoyed this webinar and appreciated each of your excellent talks. So I think, to encourage everyone to go forward and be good team leaders and good team members as well on your various teams. So thanks again. Thanks a lot to Beth for organizing for us.
Thank you all.
Thank you.
Thank you. Thanks for the invitation.
Bye-bye.
Thank you.